So I'm Kurt Thorn, and I'm going to talk now about part 2 of our lectures on digital image analysis. And specifically, I'm going to cover now some basic image analysis techniques and algorithms and tools that go into just about any kind of image processing you're likely to do. So just to refresh your memory here, we talked about this in part 1 of this lecture, but a digital image is nothing more than many measurements of light intensity in your sample. And so you have this image, it's got many points in it. You can zoom in and see the individual pixels that make this up. And each pixel here is just represented by a digital number that tells you how bright it is. And in this image here, they go from 0 to 255. And so most digital image analysis works on these underlying numbers to do mathematical calculations that will enhance or suppress certain features in your image or extract information from it for you to analyze. So I first want to talk a little bit about how you would normally correct an image for common microscope artifacts. Artifacts isn't almost the right term, but just things that go into your image that make it not quite as linear and as uniform as you'd like it to be. And the first one is background correction. And any image you'll take will have background from various sources. Some of it is purely mathematical in a sense because your camera has a non-zero offset. So your camera doesn't read 0 if no light goes to it. It always reads some finite positive number. It's usually a few hundred or maybe 1,000. And so you'll never see intensities below that in your image. But then you can have real background, physical background, which comes from media autofluorescence, background fluorescence in your sample, stray light in your room, the fact that your room's not totally dark and you're picking up room lights in your image. And ideally, if you're doing an image analysis, particularly if you're doing a quantitative image analysis, you'd like 0 to be meaningful in the sense that you'd like 0 to reflect what you see when there's no real fluorescence from your sample. So that if there's no fluorescent protein in your sample or no antibody staining, you'd read 0 and not 200 or 1,000 or whatever. And so generally the way you deal with this is background subtraction. So you try and measure or estimate your background in your image somehow, and then you subtract that off. One way to do this is taking a camera dark image, so take an image where no light is coming to your camera, and then subtract that off. Or you could take an image of, say, unstained sample or a mock stained sample that doesn't have any dye in it and estimate what the background intensity is there and subtract that off. And then there's also approaches where you can estimate the background from the image, and that's what I'll mention first. So here's an image of a cell. There's actually, one bright cell here and a sort of dim cell over there and another dim cell over here. And if we plot the pixel intensity histogram of this image, so here we're just plotting on the y-axis the number of pixels with a given intensity, and on the x-axis that intensity. You can see the intensity here goes up to about 10 or 20,000, or I guess 30,000. And this image here, you can see is mostly background. It's mostly this dark background. And so the histogram is dominated by this huge peak here, which represents all the background pixels. And then the cells are this sort of tail on this histogram and this little bump right down here. But since most of this image is background, it's pretty easy to figure out what that background value is. And if we zoom in on that peak there, you can see the peak here is centered around 1,200 or so. And so we could work out, say, the modal value in this image, or we could fit a Gaussian to just this peak, or look at the median or some other statistical measure, which will basically tell us what that background value is. And now we could subtract that off from every pixel in our image, and then we would measure roughly 0 for where we don't have any real biological fluorescence. So this is a very commonly used approach to estimate background from images. You can do more sophisticated things where you look in local windows, so you don't use a single background for the whole image, but you try and estimate what the background is in different regions independently in case your background is non-uniform. But if you have good images with a pretty uniform background, this very simple method works quite well. As I mentioned, you can also acquire a dark image, and this is actually very useful because it tells you something about your camera and your instrument background. And this you do by just blocking light going to the camera. You close a shutter, or you even take the camera off and cap it, and then you just take an image. And this allows you to measure both the camera offset and its dark current and read noise. And then it also gives you an estimate of what your real minimum background is, your true background when no light is arriving at the camera. And this can be very handy because it lets you figure out what's real and what's autofluorescent. So if you see your image and you measure a value of 1,000 in the background, it's hard to know, is that because I have bad autofluorescence or because my camera just has that offset? And so if you take your camera off and you measure an offset of 200, then well, gee, there's 800 digital numbers here above that offset that must be coming from autofluorescence or stray light or other things that maybe I can get rid of. This is what a dark image looks like. This is what a good dark image looks like. If you have a cheap camera or one that has problems, you'll often see that it's not so uniform. But an ideal dark image looks sort of like this. It's just this featureless gray image with a little bit of noise on it. And so you could then subtract this off from your images, and that would correct for this camera offset. In general, if you were going to do that, you'd want to acquire a number of these and then average them to minimize the noise in there. Another very common source of error in your image that comes from microscope aberrations is what's called a shading correction or non-uniform illumination. And this just comes from the fact that it's very hard to get truly uniform illumination over your sample. And so you may see that pixels in the center of your sample are brighter than pixels at the edge. And in general, you'd like to correct for this too. You don't want to bias your results by measuring brighter cells in the center of the field of view. And so if you have some samples that have a lot of cells in the middle, they'll read as brighter than samples that have a lot of cells at the edge of the field of view. And correcting for this is also quite simple. You just image a uniform fluorescent sample. And commonly what we use are just pieces of fluorescent plastic cut to microscope slide size. But you can also use uniform solutions of fluorescent dye suspended between a cover slip and a slide, or any other sample that's fluorescent and will be very uniformly distributed. And so if you take a picture of one of these samples, you usually see something like this. Generally it's brightest in the center and it tails off as you go to the edges. Sometimes the brightness is not centered, the bright spot is at the corner or whatever. But it looks something like this, and usually the variation intensity is around 20 or 30% over the field of view. And so you can divide your image by this to correct for that non-uniformity in the illumination. There are also generally non-uniformities in the detection, and that's sort of all rolled together here, but for most purposes, that's an okay assumption to make. And so if you put these both together, this is how you would do that analysis. So you would see, you measure some image here, that's this IMES, and it's equal to your true image in the absence of these problems. The true image times the shading correction. So your true image is multiplied by whatever this non-uniformity is, and then you add to that this dark image. And so just doing some algebra, you can get out that your true image is equal to your measured image minus the dark image divided by the shading image. And in general, this is something you probably want to do on your images if you're trying to do very quantitative imaging, or if you're doing things like image stitching or tiled images where you want to make sure that they don't have any checkerboarding, they have a very uniform intensity across the field of view. But in general, this is a good procedure to do. And almost any software package should be able to do this kind of simple image math where you're subtracting one image from another image or dividing one image by another image. You do need to pay a little bit of attention here that in general you'd like to normalize your shading image such that it goes from, such that its mean is 1, so that you don't change the intensities when you do this division. And you also therefore need to make sure that you use what's called a floating point format to store this image, that you don't store it as just integer numbers, 0 to 255 or 0 to 65,000 or whatever, because then normalizing this to 1 will make it all zeros or ones. You want it to have floating point, which means it has decimal places in it, so that the value can be 0.99 or 1.02 or whatever. But pretty much any software package should let you do that. So that's the first thing I wanted to talk about, are just these basic image corrections. And now I want to get more into image analysis or image processing, where you're applying manipulations to your data to try and enhance or suppress certain features. And an extremely common and very powerful technique is what's called digital image filtering. And the idea here is you have what's called a kernel, so this little matrix of numbers here. And at every pixel in your image, you go and apply this kernel and calculate some product between the kernel and that portion of the image, and then replace that pixel in the image with that product. And so the way this works is here we've got a little 3x3 square here. And the idea is if we wanted to apply this to a single pixel in the image, we'd center this 3x3 box on that pixel in the image, and then go around and take the pixels around it and multiply them by these numbers, add them all together, and then replace the central pixel with that value. And so what this particular kernel does is it does a smoothing or an averaging filter. And so it takes this 3x3 neighborhood and replaces each pixel with the average of its neighboring pixels. You can also do that with what's called a Gaussian smoothing kernel, where instead of being just a uniform set of values over the kernel size, here we weight the center more highly. We weight these in a Gaussian fashion, so we weight the center highly and then roll off towards the edges. And how this works is, here's, say, a little 5x5 image we want to filter. So we take our 3x3 kernel here. We then drop it on this 8 here. And so now we go through one by one, and we say, OK, here's 10 times 1 is 10. 11 times 1 is 11, and so on. And we multiply our way through and add these all up. And eventually, you get the value 14 out of that. And so you would go in here and then replace that original 8 with 14, because that's the average of these 9 pixels. Here's another example of this where we've got a little more complicated image. So we've got this image here. And again, we want to use the simple 3x3 smoothing filter on it. And here's what the digital values in that image are. You can see there's these values here that are 113, which correspond to this gray line here. And then these values that are 255, which correspond to the white diagonal line there. And we're going to put the filtered values over here. So the idea is we take the first corner of this image, drop that smoothing matrix on, and then multiply and average those guys. And that gives us a value here. Do it 1 over for the next value, and so on and so forth. And if we do it for the whole image, we get this, and that looks like this. And so you can see we've averaged out and blurred these structures here. One thing, if you were looking at this closely, you may wonder about is how do you deal with these edges, right? We're applying this 3x3 kernel here, and so if we want to filter something in this top edge here, we need to figure out what the numbers up here, one row above that edge, should be. And obviously, there aren't any numbers there. And so there's a number of different approaches that people commonly use to deal with this. The simplest is probably just to assume that any numbers outside of your original image are 0. But equally, you can generate those numbers by taking this top row and duplicating it, or wrapping around so that the top row is connected to the bottom row, and then you would get your numbers from there. And I think that's what was done here. The net effect of this kind of kernel is to do a smoothing or an averaging of your image. And as I said before, you can also use a Gaussian kernel, which looks like this. And if you look at what it looks like this. And so it's basically what you would expect is bright in the center and then falls off smoothly as you move out from the center. And that would do a similar kind of blurring operation, except it would be more weighted just towards the pixel in the center and its nearby pixels. So why do you want to do this? There's many reasons you might want to do it. You might want to generate a blurry image of your cells or of your data just to smooth out small little artifacts, or to just estimate what the mean brightness of something is, not dominated by small things. But one very interesting reason is because if your image is sampled appropriately at what's called the Nyquist limit, and the camera lecture talks about this, the point spread function of your microscope, so the minimum resolvable element in your microscope, will be spread out over many pixels. And that means that the information in adjacent pixels is correlated with each other, and they're not completely independent. And there's some redundancy there. And if you use deconvolution, you take advantage of that redundancy in a sort of mathematically correct way to try and reconstruct a more accurate image by using that redundancy to infer what the underlying data that gave rise to your image was. But even if you're not doing deconvolution, you can exploit some of that redundancy by smoothing with a Gaussian kernel that's sort of the same size as your point spread function. And part of why that helps is because since there is redundancy in your image, if you have a single pixel that's really bright and everything around it is dim, you know that can't be real because any real bright object in your sample would be spread out over multiple pixels. And so a single bright pixel like that has to come from some kind of noise artifact. And so smoothing will help suppress those artifacts and give you a better representation of what your underlying image looks like. And so here's an actual PSF. So here's a point spread function measured from a microscope. And here's that Gaussian kernel again. And you can see they do look fairly similar. And in fact, if we took a little bit bigger Gaussian, it would be an even better match to this. And so the idea is that this is a good approximation to the underlying distribution that our microscope generates. And so smoothing by this will remove any sort of nonsensical hot single pixels or bright single pixels that couldn't have come from a real object, which would instead generate a bright thing that looks like that. And so here's a kind of artificial example where we've taken an image, a noisy image. This was actually a computer-generated noisy image, but the principle is the same for real noisy images. And you can see there's structure in here, but it's kind of hard to see it. And when we do this Gaussian smoothing filter, you can see these structures are now easier to see because we've averaged out some of the noise, but we haven't averaged out the real structures by as much because they're about the same size as our averaging filter. Here's a bigger example. These are real microscopy images. It's a little harder to see here, but you can see here's the original image, and then on the bottom here we've filtered it with a Gaussian filter. And again, it's a little bit easier to see structures in there, particularly if you zoom in on it. However, these kernels can be used for many different kinds of operations, not just smoothing. Another very common one is edge detection. And so you can sort of see what we're doing here if you just look at these filters. So there's two different filters here, one on the top and one on the bottom. They do essentially the same thing. They take the difference between one row, so this top row here, and then the row two away from it. And so if there's a bright thing in this row and a dim thing in this row, we'll get a bright output. But if these two are about the same, you'll get a zero output. And here we're doing the same thing, but we're just now biasing a little bit to be more centered around the pixel vertically. Take more information from the vertical pixel in the center there and not the ones to the left and the right. And so what these things are going to do is enhance edges that run horizontally through our image. And in fact, if we apply that to this image here, we apply the top filter to this image, here's what you get. You see that these actin filaments that are running roughly horizontal along here jump right out. The ones that are running vertical are totally suppressed. And the ones that are running at a diagonal sort of show up somewhat. You can equally well rotate each of these filters 90 degrees and get a set of vertical edge detection filters. And if you apply a horizontal filter to the image, a vertical filter to the image, and then add those two images together, you get kind of a detection of all edges regardless of orientation in the image. So this can be very handy for finding boundaries of things, any type of sharp edge that you want to bring out in your image. And then there are similar sorts of filters to that edge detection where instead of looking for edges, you're now looking for just bright objects surrounded by dim objects. And these are contrast enhancement filters of various sorts. You can see here's one here where we're taking a very large value times the center pixel and then subtracting off a small amount of information from all the neighboring stuff. And there's a number of different varieties of these. They go by different names and have slightly different shapes, but they all follow this general principle of a bright central object and then subtracting off its neighboring pixels. So these are called things like unsharp masking or unsharp filters. That's a sort of old technical photography term. Laplacian filters or Laplacian of Gaussian filters. They all do basically the same thing. They differ in the details, but basically all do this contrast enhancement. And you can see if we apply that to this image again, that really makes these actin filaments pop out, but at the same time it also highlights sort of speckly noise and stuff in this image. So these are both good and bad depending on what you're trying to do. They tend to enhance noise, but they also enhance contrast in your image. You can also use nonlinear filters. So everything we've talked about so far has been a linear filter, meaning the output is a linear function of the input. It's just generated by multiplying pixels by the kernel values and then averaging. But you can also do nonlinear filtering where you do things like median filtering. You take a box, a 3x3 box, say, you pick out the median value of those 9 pixels and then replace the central pixel with the median of those 9 pixels. These are have different properties than the linear filters. One thing they're good for is smoothing while maintaining edges in your image. They're also very good at removing hot pixels or other single pixel garbage in your image. So that's what I wanted to say about filtering. And then the next topic I want to talk about is thresholding. And thresholding is an extremely common technique for identifying objects in images. And so the idea here is if you look at this image on the left here, you can see there's a bunch of nuclei in it, these bright white objects. And then there's a lot of background. And we'd like to automatically separate these nuclei from the background, automatically identify where they are and which pixels are in the nuclei as opposed to which pixels are in the background. And so the idea here is if you look again at this pixel intensity histogram, so here's the histogram. This time it's plotted on a log scale so we can see the dimmer information better. Again, like that first pixel intensity histogram I showed you here, it's dominated by the background. There's this huge peak right here of the background data, all those pixels that are black and in the background. And then it very rapidly falls off. And then you see this long tail, which eventually goes away. And that corresponds to the white objects here, to the nuclei. And so the idea behind thresholding is that we can, by just picking an intensity somewhere in this range here, in between those two objects, we can separate this into foreground and background, into nuclei and background. And so the idea is we would say maybe take everything above this value in this red box here. And now if we just color those pixels red on this image, you see we've picked out all our nuclei. And for images like this, where there's very good separation between foreground and background, between the objects you're trying to segment and the background, this can work extremely well. As you can see here, it basically works perfectly. The bigger question then is, how do you do this automatically? And so here's a little bit of a tougher case. This is actually not a microscope image, but a dot blot. And you can see there's some really dark objects here, and then some objects that are clearly there, but they're not a whole lot darker than the background. And so setting the cutoff here matters quite a bit. If you set it too high, you miss these dim objects. If you set it too low, you start picking up a lot of background noise. And also, you would like to be able to do this threshold choice automatically, or at least in a sort of unbiased, objective way. And so this is just a screenshot from image J here from their thresholding. You'll see there's this auto button here. And if you use that auto button, it automatically picks a threshold. And this is the threshold it picks. You can see it does a pretty good job. It gets most of the dark objects, but not quite all of them. And how does this work? So it turns out actually in image J, there's many, many different algorithms it can use for doing automatic contrast or automatic threshold generation. But the most common one you'll see used probably is this, what's called Atsu's method. And this basically works by assuming your image is made up of background pixels and foreground pixels, and that those are both roughly Gaussian distributed. And it looks for basically the minimum between those two distributions and then sets the threshold there. And so here's an example from a paper of just trying to segment the DNA in this mitotic spindle, and there's what the DNA image looks like. And here's what Atsu's method detects for the appropriate threshold. And this works pretty well. There are many other, I think the ImageJ threshold tool has something like 15 or 20 threshold algorithms. They work better on, some work better on some kinds of images than others, basically because of the assumptions that are built into them. There's a lot of refinements on this for doing more sophisticated ways to threshold. Image segmentation after thresholding, where you try and separate objects that are touching. But at the base of all these segmentation algorithms is this idea of thresholding, that you can separate your picture into a foreground intensity and a background intensity in some automated manner. So one problem with this approach I just want to mention, assuming, say, we wanted to quantify nuclear intensity by this thresholding approach, is that it's biased towards brighter objects. And so ideally, if you're trying to quantitate something here, you might miss objects that don't have any fluorescence at all. They might be there, but you just can't see them, or they're so close to the background they don't get picked up by your thresholding. And so you would bias your output data. And so ideally, you'd like to use a second channel you're not quantifying on to independently define the objects to measure. And the idea here is you'd have some channel that uniformly labeled all your objects to detect, independent of the thing you wanted to measure, so that you could, in a rigorous and unbiased fashion, pick out all the objects. And then inside those objects that you detected, you would just quantify this other signal. The other thing that thresholding does is it gives you a binary image out. It's 1 inside your object and 0 everywhere else in the image. And so of course this can be used to identify objects. You just find all the things that are 1. But this can also be then manipulated to further separate or define where objects are. And I just want to touch on this in a little bit of detail. This is, again, another large area of image processing, of binary image processing. But I'm just going to mention a few very common techniques here. And the 2 common techniques are erosion and dilation. And superficially, these look a little bit like the filtering approaches we had before. We have a kernel here. It's now called a structuring element. We have our image here. It's now just 0s and 1s. And it's the image of this little funky-looking object here. And the idea here is we are going to apply the structuring element to this image and either take only pixels where the structuring element completely fits inside this object, or we're going to create new 1s in this image wherever there is a 1 that matches the center of the structuring element. So the idea in erosion is that we want to go in and inside, at every 1 in this image, we put the center of the structuring element there. And if every 1 in the structuring element overlaps with a 1 in our binary image, then we keep that pixel as a 1. Otherwise, we set it to 0. So if we would put it down here, we'd have 1s that overlap these 0s. And so we'd set this pixel to 0. Go here, we have 1s that would overlap with 0s. We set that pixel to 0. But if we put the structuring element here, you can see that 3 by 3 box of 1s would all match 1s in the image. And so we'd keep this pixel as 1. So if we do this erosion here, we get an image here that now just has 2 pixels in it that are set to 1. And these are just the 2 pixels that are fully inside the body of this object here. So there are these 2 pixels in there. So erosion is a good way to get rid of sort of stray pixels, little things that stick out from objects. And then its counterpart is called dilation. And the idea in dilation is we go and we would drop the structuring element on here. So we'll drop the structuring element here and match the center 1 with this one here. And then we would set all these 0s to 1s wherever there's a 1 in the structuring element that overlaps with this image. So if you do that to this guy here, you do the dilation, now you get basically a big box here that corresponds to dropping that structuring element on these 2 1s. And the net effect of this, like erosion followed by dilation, is essentially a smoothing of this image here. We've removed these kind of pointy little small structures that stick out and left with just a smooth representation of it. So erosion followed by dilation is a very common way of smoothing boundaries in binary operations or in binary images. There are a large number of other binary operations. If you go into a program like MATLAB, there's probably 20. So you can find automated versions of sequential erosion and dilation for doing smoothing. There are operations like hole filling. So if you have 0s completely surrounded by 1s, you set those 0s to 1s, which is very nice for making contiguous closed objects. And then there's things that will do stuff like removing objects at borders, which is very useful for removing data that's at the edge of your image that's partially cut off that you wouldn't want to quantify. So along with filtering, these binary operations are kind of a real workhorse in digital image analysis. And that's really all I wanted to say today. This is just designed to give you a taste of the tools that go into digital image analysis. Again, thank Nico Sturman for providing many slides that I used here. And then also just list a bunch of books that have a lot more detail on these kinds of approaches that you can look into in the future. Thank you.